All right, well, let's get started. Thank you for being with us this morning. My name is Rick Dantzler, and I'm the Chief Operating Officer for the Citrus Research and Development Foundation. This event is an annual event that we sponsor with Florida Citrus Mutual to help update growers on some of the latest projects that we're working on and some of the new things we are learning. Normally, we have this meeting live in Bonita Springs, but of course, with COVID, we've been having to do it virtually. We did that last year, and we're doing it again this year. Let's hope that next spring we'll be together again down in Bonita Springs. You know, the citrus industry has a long history of taxing itself to provide research funds to help answer the questions it believes need to be answered to help it grow a good and profitable crop of citrus. CRDF has been the recipient of those funds since 2009, and we have funded 462 projects. Much of that funding has come from growers, and much of that funding has come from the state legislature. We are a not-for-profit corporation, so we are not allowed to lobby. We have to count on Florida Citrus Mutual and growers to do that on our behalf. And I'd like to thank Mike and his crew for the great job they've done over the years, making sure that the citrus industry receives the funding that it needs to stay profitable. I know that it has been a hard year, but I am confident that next year is going to be much better. We had a tremendous crop set. The bloom was wonderful. We have some great research projects in the pipeline, and I think things are going to get easier, and that next year is going to be far better than it has been in the last year. There may be a number of legislators on the line. If so, I want to especially thank them for the funding assistance they give us. One of those legislators is a citrus grower himself, Ben Albritton. I don't know if Ben is with us or not, but thank you, Ben, for your assistance in helping the industry secure the funding it needs over the last number of years. We have a great program in store for you today. We decided to focus on field trials. The reason we did that is because that is where an awful lot of the energy and frankly, the funding has gone in the last couple of years. So we thought it would be good to share with you the fruits of those labors. One of the things that field trials gives us is an opportunity to find out if what we have learned or what we suspect can hold up in a commercial growing environment. In fact, that's exactly why we do field trials. And 3 years ago, the industry came up with a really big idea, and that was to see if we could plant 5,000 acres of large-scale field trials to find out what worked best. This was a great example of growers helping growers. And with that big idea, Craft was born. And here, a little over 2 years after it was created, Craft has 103 projects in the ground. Those projects span the entire breadth of the major citrus growing regions in Florida, and in my view, has been a tremendous success. Much of that success lies with the board, with the technical working group, with Kristen Carlson, with Tamara Wood. All of those persons that have been involved with Craft have helped create a tremendous program. And I am convinced that when the data from these trials begins to come in, we're going to see what a great program it has been. So to those who have had a hand in the creation of Craft and getting it off the ground, thank you very much. It has a significant amount of scientific rigor connected to the projects. That was one of the concerns at its inception, but I think the technical working group and the board have done a great job making sure that what we get from these trials in terms of data is going to be data that can help you in your citrus operation. So today what we're going to do is hear from 4 of those grower cooperators about their Craft trials. In between, we are going to hear from 5 of the great scientists that CRDF has helped fund over the years, and they are going to share with us what they are doing in terms of field trials. Each scientist is going to be focusing on a specific and different thing, and I think you're going to find it very enlightening. One housekeeping measure that I need to get out of the way, we hope that this will be interactive. So if you have questions, please hit the private chat icon at the bottom of the screen and send your questions to Tamara Wood. Tamara will ask the questions on behalf of all of you. I think that will be more orderly. And we want to take the questions as the presentations come up. So if you have a question about a particular presentation that comes to mind during that presentation, send that question to Tamara and we'll entertain it as soon as the presenter finishes with his or her presentation. Without further ado, let's get started. Our first presenter today is Dr. Sarah Strauss. Dr. Strauss received her PhD in plant biology from Arizona State University. Her dissertation was on mechanisms and patterns of soil carbon and nitrogen recycling in 2 extreme environments, which I think sounds like perfect training for what she has been working on behalf of the Florida citrus industry. We are lucky to have her advising us on matters involving cover crops, which is what she is going to speak with us about today. So with that, Dr. Strauss, you're recognized.
Thank you very much. Let me share my screen here. All righty. Thank you again for that introduction and for inviting me to speak this morning. I would like to share with you some of the information that we have learned in the last 2.5 years of doing some research with cover crops in commercial groves, and go through some of those findings and some of the things that we're still working on. So to start with, what are cover crops? Cover crops are basically crops that are planted strictly to benefit the soil. They are not harvested for profit, and they're not a new idea. This photo, it's the best I could do on the scan, but it's, if you look at the caption, it's from 1929. Planting, in this case, some mustard cover crops in a citrus grove. So this is not a new idea. But for a variety of reasons, it is not as common in citrus or in other crops right now. That said, it's becoming increasingly common, again, in grains, cotton, corn, soybean farming, vegetable farming, particularly in areas in the Midwest, where they're planting their cover crops during their fallow season. And then they're often combining those cover crops with other management practices like conservation or no-till management practices in an effort to, again, really boost that soil health in those particular crops. But why plant cover crops? There are a variety of potential benefits in addition to this idea of soil health. Cover crops can certainly help reduce weeds, which is obviously a big issue here in Florida. It can help reduce erosion and reduce soil compaction by having plants in the ground, having that biomass below ground that helps retain your soil, prevent it from eroding away, and it helps reduce compaction. And one of the big benefits of having cover crops is the potential to increase your soil organic matter. And by increasing soil organic matter, you can then lead to changes and enhancement in soil nutrient cycling, potentially nitrogen availability, which could lead to benefits to your tree. In addition, having increased soil organic matter can sometimes also help increase soil moisture, which has potential benefits as well. But how do cover crops increase soil organic matter, and how might that impact the citrus tree? So soil organic matter is material that comes from living organisms, plants, animals, microbes. And microbes play a really critical role in building this soil organic matter, which is why, as a soil microbiologist, I've been looking at cover crops. And so when that material is added to the soil, it's basically serving as resources, food resources for microbes in the soil, and it allows an increase in their biological activity, potentially changes in which organisms are present by having increased organic matter. And having an increase in that biological activity can then lead to changes in decomposition, the amount of nutrients that were released, how those are cycled through the soil, and ultimately potentially influencing your crop and influencing the plant growth. And so while yield is certainly one of the key parameters that we're all interested in, it can take time for some of these changes to occur in the soil. And so therefore, to try and gauge whether we're on the right track before we might see yield changes, we're often making measurements of some of these other components. So looking at soil organic matter itself, looking at biological activity, looking at microbial activity, looking at nutrient changes, and seeing if we see patterns with those, which we hope will then lead to changes in the crop itself. So there are a wide number of cover crops that can be planted, different species, different mixes. Some of the ones that I've listed here are things that we've been trying in some of our field trials. Perennial cover crops, a lot of folks, when we think of cover crops, particularly here in Florida, we think of perennial peanut as it grows really well out here. That can have both nitrogen and nematode benefits. Bahia grass, we don't always think of it as a cover crop, but if you're planting it purposely into your row middles, you could consider that as a cover crop. That provides some nice below ground biomass. And then there are a variety of annual cover crops. And so we've looked at a variety of different legumes, cowpeas, sun hemp, vetches, and there are also a variety of non-legume plants, rye, wheat, a variety of millets, sorghum, Sudan grass, daikon radish, that all can provide some of that great biomass that is really important for building soil organic matter. In our trials in particular, we've had great success with sun hemp. Unlike other parts of the country where they're doing cover crop research, we can grow sun hemp year round very well in Florida. And it's a legume, it fixes nitrogen, and so that has a potential benefit. We've also seen great success with daikon radish, planting that in mixes in the winter. It does not like our hot summers here. But you can see in the photo, it can create quite a bit of below ground biomass, which again is great for building that soil organic matter. There's been a lot of research, primarily with row crops, on the differences and the potential pros and cons of planting a monoculture or only planting one cover crop species versus a polyculture, planting multiple species. And a lot of those pros and cons and which might be best for you also depends on some of your goals and management. Establishment time is one of the big differences. As we probably know, perennial peanut can take a little while to get established, which is why we did not look at it in some of our specific trials for this project, because it can take 3 or 4 years. Annual cover crops, however, you might start to see benefits a little sooner, because hopefully if you get a good germination, they'll get going a lot faster. There have been some work on the potential pros and cons of having greater diversity of plants and whether that changes the amount of nitrogen. That's something that I think we still need to look into a little bit more closely, particularly for our Florida soils. So for this field trial that CRDF has funded, we wanted to get some quantitative data on the impacts of cover crops in Florida groves. So there's been a lot of talk about it. There have been a number of folks that have been working with it, but we wanted to get some numbers. And so we have 2 trials occurring in commercial citrus groves, 2 different groves. For time, I'm just going to present the results from one location today. However, we are seeing similar trends at the other site. In this grove, they are Valencia on Swingle. The trees were planted over 20 years ago. And this is taking place down here in Southwest Florida on the Flatwoods. And so along with my collaborators, Dr. Davy Kade-Kompeni, Dr. Ramdas Kanasari, and Dr. Tara Wade, we've been measuring a variety of different parameters, looking at tree and root growth, looking at fruit yield and quality, looking at weed growth, some of the economic benefits, and then my group has been focused on the soil microbial community and how that relates to soil organic matter and nitrogen. So a couple of the things about these trials, since these are down here in Southwest Florida, the trees are planted on bedded rows. And so the cover crops are only planted on the raised row middles. They're not planted in the swales. So these trees are really only seeing cover crops on one side. Before we've been planting the seeds, we have been herbiciding the row middles and then waiting a couple weeks. This gives a nice clean slate for those cover crops and reduces some of the competition. And we found that that really helps increase our germination chances and our coverage. We've been using a no-till drill to plant our cover crops. There are a variety of methods available. And unlike with row crops, management for cover crops and citrus is a little different, particularly when we're talking about annuals. So we, in our trials, had two different cover crop mixes. One mix has legumes and the other mix only has non-legumes. So the mixes are basically the same except for the presence of legumes. And we really wanted to try and get an idea of what is that legume contribution to some of the benefits of a cover crop. The other is that unlike in row crops, our goal is not to fertilize or irrigate these cover crops. And we also want to allow them to flower and go to seed to build up a seed bank. And so that's another consideration when we're thinking about what mixes to plant. One of the most critical things that we've learned, though it seems kind of obvious in the last 2.5 years of planting cover crops, is that the timing of planting these is really critical. Really making sure that you get them in the ground while it's raining. And so we've been doing 2 plantings a year, one at the beginning of the rainy season at the beginning of June, and one towards the end of the rainy season at the end of October, early November. And that's been our practice for these particular trials. So what have we seen in the last 2.5 years? So one of the first results that I want to share with you, and I think one of the ones that I personally was really excited to see, is changes in soil organic matter after just 1 year of cover crops. And so in the next couple of graphs that I'm going to show you, we've got the time of the trial on the bottom. We have our legume plus non-legume mix in green, our non-legume mix in orange, and the grower standard control in blue. And so you can see after just 1 year of cover crops, we had a significant increase in soil organic matter in both of the cover crop treatments, and that we maintained that increase throughout the 2.5 years of the project so far. One thing that's been seen in a lot of row crop studies is that some of the biggest increases in soil organic matter with cover crops might occur in that first year and that first initial planting of cover crops. And that's certainly what it looks like we're seeing here. And that after that initial increase, the further increases might take a little longer. And so that's what we think is going on here, but we're encouraged that we have consistently seen greater soil organic matter in both of these cover crop treatments in the last 2.5 years. We're also seeing changes in soil nitrogen, and again, particularly in the cover crop mixes with legumes, which is not too surprising. It's a significant increase in ammonia in the soil in those legume plus non-legume mixes. And we're seeing that again over in the last 2 years, and that's been maintained. Measuring soil ammonium nitrate can be a little tricky. The amounts can vary depending on when your samples are collected and the relationship with soil moisture. And so we think that might be one of the reasons why we're seeing some variability here. But again, we're still seeing this trend of greater nitrogen in these mixes that have the legumes. And so those things we think are both contributing to the big change that we're seeing in the microbial community. And so in this graph, I'm showing the total bacteria and archaea abundance. And so we see a significant increase in the first year, and that continues in the second year, and that increase is growing. We're seeing a bigger difference in the total change in bacterial abundance with cover crops, both cover crop treatments after 2 years. And not only seeing increases in the numbers of bacteria and archaea under cover crops, but we're seeing changes in which organisms are present. In particular, if we're looking at organisms involved in nitrogen cycling, since that's a nutrient that we're all potentially very interested in, we see changes in the organisms involved in nitrogen fixation, particularly in both cover crop treatments, but particularly in those treatments that have legumes. This is again not surprising, as legumes have a symbiotic relationship with bacteria that fix nitrogen. And again, this effect appears to be cumulative when we're seeing increases in these numbers over time under these treatments. We're seeing similar patterns with nitrification, which is the conversion of of ammonia into nitrate. Again, seeing significant increases in organisms involved in those processes under both cover crops. But again, more increases in the treatments that have the legumes. Again, there's more nitrogen cycling in those treatments. So it's not too surprising that we're seeing more organisms that are capable of cycling that nitrogen. Same deal with denitrification. This is the process of converting nitrate through a number of different processes and a number of different organisms, ultimately into N2 gas back into the atmosphere. And again, in cover crops, we see more organisms capable of these nitrogen cycling processes and denitrification. So all of this leads us to think that, again, we are seeing a significant change in the microbes and the microbial functions when we are planting these cover crops. And we're seeing bigger changes, particularly related to these nitrogen cycling functions when we plant legumes. We're also seeing changes in weed pressure. This is work that Dr. Ramdas Kandasari has been doing. These graphs are a little different. But again, if you look at our legume plus non-legume mix and the non-legume only, green color here are the number of cover crops per square meter, and blue is the number of weeds per square meter. So you can see in the cover crops, we have primarily cover crops. None of the grower standard control, we have mostly weeds. And we see the same pattern both in 2019 and 2020. As I said, we're in about year 2.5 of this trial. Our next big sampling point actually occurs next month in August. And so a lot of the measurements that we've been making occur annually. There, as you saw in the graphs, a couple of them have occurred at slightly more frequent intervals, depending on what that measurement is. So we should have, and I'm very excited to see what data we get next month to round this project out. So a quick summary, we have not seen any changes to the yield, tree canopy, or roots after 2 years. How long will it take to see changes in the tree? That's a little hard to estimate. As I said earlier, that's one of the reasons we've been trying to look at some of these other parameters to see if we're on the right track. Row crops often estimate taking at least 3 years of cover crops before effects are seen. So we're sort of getting into that time frame. But again, that's with row crops. Trees are, as we all know, are a slightly different animal. And so it's hard to say at this point. But again, based on these changes that we're seeing these increases in soil organic matter and microbial abundance when we're planting these mixtures of annual cover crops, that we're seeing these changes in soil microbial nitrogen cycling, to me indicates that I think we're on the right track. One of the big things, as I said, is the timing of planting is really important for establishment. And the other sort of side benefit that we've realized is that planting cover crops reduces your mowing frequency. As I mentioned earlier, we're really wanting these cover crops to flower and go to seed, and so that can take a little time. And so we're not mowing these cover crops once they're planted. Certainly during harvest in the spring, they get trampled a bit, but then some of them bounce back up, or we're planting the next round shortly after harvest. And so these rows have not been mowed since we started this trial. Of course, there are a large number of things that we're still very much interested in. And I think, as any good field trial and experiment does, it just generates additional questions. And so one thing that we're starting to work on a little bit more is working on optimizing our mixes, trying to really dial into how to time these different varieties with each other and for our Florida soils. We started some new projects recently to do some additional measurements to try and get a better understanding of exactly how these cover crops are contributing to soil carbon and nitrogen. Certainly we're seeing changes in the nitrogen cycle, but there's some other measurements that we would like to make to try and have a better idea of the availability over time and availability of the trees. And I've recently started some new trials with some other colleagues on ridge soils. So we'll be planting cover crops on both sides of the trees and with younger trees. And so we're hoping that those two factors might lead to faster changes in the tree parameters themselves compared to these older trees that we've been working with in these current trials. So with that, I'd like to thank my collaborators. I'd like to very much thank Dr. Antonio Castellano-Hinoso, who's the postdoc in my lab and who's done a fantastic job with all the microbial work. And I'd especially like to thank CRDF for funding this project. There is also still a cover crop grower survey, and I can put this link in the chat when I'm done, that Dr. Tara Wade is still looking for folks to fill out. You do not have to currently be growing cover crops to complete the survey, but we would appreciate your input. That I would like to thank you very much.
You bet, Dr. Strauss. Thank you very much. I know we have a couple of questions. Tamara, would you like to share those?
The first question that came in was, is using compost as effective as using cover crops? And is it recommended to do both cover crop and compost?
That is an excellent question. So they're sort of two different, ultimately both of those practices are trying to increase soil organic matter. Cover crops, by having biomass, by having that plant in the ground year round, it's a bit of it's a more established way, I think, of lack of a better word, of building that organic matter. Compost is sort of a one-time application, or if you're lucky, a couple times a year. Cover crops are contributing to that soil organic matter all year round. We have done some small trials here at the research center trying to look at applying compost and cover crops in the row middle together. So applying compost and then planting the cover crops. We actually haven't seen really big changes in soil organic matter by doing that. We do have some trials that are starting out where we've been applying compost. Actually, there's another CRDF funded project looking at compost application initially, and then we're going to be adding some cover crops in later. And so I hope to have some more solid numbers on those differences for you in the future.
Thank you. The next question is, at one time, hairy indigo was planted in groves as a cover crop. Other than its invasive nature, are there other factors that would or would not make it a candidate for utilizing as a cover crop now?
That is a good question. I'm not 100% certain on all of the different issues with hairy indigo, to be honest. I know it's been talked about in planting in various places. Certainly there are a number of, I think as Dr. Conassary might tell you, one man's weed is another person's treasure. And so how we view some of these species really depends on our purposes. And so, there are certainly some things that we might want to look into if they grow well here in our row middles. I'll have to get back to you exactly on some of the details with hairy indigo.
Thank you. The next question was, what was the drop percentage on the crop this past season?
We didn't see any changes in yield with our treatments. I would have to go back and look at the numbers to see just yearly trends in the grower standard control, but we did not see any impact of our cover crop treatments on yield.
Okay. Next was, what was the grower standard control? Was it bare soil, natural occurring weeds, or something else?
Naturally occurring weeds.
Okay. What are the predominant plant species in the grower standard? What size are the treatments?
I'm not of the weeds.
I believe so.
We'd have to talk to Dr. Ramdas Conassary. He's got counts and data on specifically what weeds are present in those grower standard controls. I'm not 100% certain off the top of my head.
Okay, and we have two more quick questions. The first one is, since the cover crops are planted in the middles, how far would you expect the effects of the cover crops, if any, to extend into the rows of trees?
That's a good question. These are pretty old trees that we're working with here. And so there are citrus roots in these row middles. We find them when we're collecting soil samples. And so that's why we think that even though these are not extending all the way up to the trunk, they hopefully are potentially benefiting those trees because the roots are extending all the way through those row middles. I think that's one thing that we're very interested in looking at in some of our newer trials with younger trees that may not have as extensive root growth yet.
Okay, and we have two more, and then after that, if any more come in, I will forward them to Dr. Strauss so that she can respond to those directly. But the last two, what was the cost for these trial cover crops?
That is a good question. I'm going to direct you to Dr. Tara Wade, who's been working on the economics.
Okay, and then last, was the drop visually large compared to the grower standard control?
I'm not sure if you're talking about fruit drop, which we have not been measuring for this particular project.
Okay, thank you, Dr. Strauss.
No problem. Thank you.
Thank you, Sarah. Sarah is with UF-IFAS in the Immokalee Research Center, and I'm sure she'd be more than happy to visit with you about questions you might have regarding this or other matters she's involved in. But thank you for being with us today, Sarah. Next, we're going to have a presentation on a craft project. Before we get into these craft projects, though, I want to make everyone aware that, as you know, craft is in its infancy in terms of getting the projects in the ground. So there is not hard data that we can share with you yet. What you will be seeing is the beginning of the trial, but the data will be forthcoming, and data collection and distribution is a very robust part of the program. So I'm sure that there's going to be data made available to all growers to help you in your operation. Tamiami Citrus is an LLC in Fort Myers that has been a huge help to CRDF in the industry over its years of operation. Their principals have always been willing to serve on our committees and boards and volunteer to help the industry in any fashion. Today, we will hear from Ron Mahan, who is the Vice President of Tamiami Citrus. He is also a member of the CRDF board, and he serves as our chairperson of the audit committee. So he's had a busy year. He will talk to us about a craft trial testing brassinosteroids on a very robust trial design involving several scions and rootstocks. Tamara, please roll the tape.
Tamiami Citrus, and we own and operate the Hog Island Grove, which is under our subsidiary, North Joshua Grove LLC. Tamiami Citrus is a relatively young company, started in late 2014. We have developed and operate about 5,500 grove acres, 7,500 acres of land in 4 different locations. The Hog Island Grove is our 4th grove, and this operation will have about 1,100 grove acres on it. And we have planted this grove starting about a little over a year ago, year and a half ago. And we just completed the last phases of the planting this year. This craft, or these actually a couple craft projects, we're testing a couple of things. Brassinosteroids are a plant hormone that are relatively newly known. Just in the past several years, the University of Florida has been doing research with them, and early research it indicated that the brassinosteroids will have a positive impact on the vigorousness of tree growth, particularly on young trees, that it helps in reducing fruit drop, and indications that it also helps in trees responding to the impacts of greening. Obviously, we can't keep greening out completely, and we believe this may be a tool that can help us in helping our trees respond well to greening. So this craft project is testing brassinosteroids on a large scale, taking it from the lab to a large scale, where we are testing the effects of the brassinosteroids on these trees, which are OL4s. So you can see this is the section of the block where there's no brassinosteroids applied. And then halfway through the block, we have where the brassinosteroids start and are applied. The other aspect we're testing here is this test the brassinosteroids on the OL4s. We're also testing the brassinosteroids on B965, another relatively new, much newer scion variety. And that is over in block H2. And half the block is having brass steroids applied, just as here in F2, and half is not. So we'll be able to test both the effects of brass steroids on those two different varieties, but we'll also be testing the production characteristics of B965 against OLL4, both with brass steroids and without. Obviously, long term, our hope is that the brass steroid effector applied areas will show a much higher level of production than the areas where and prove out that the brass steroids have provided both the vigorousness of the tree growth, as well as the ability of the trees to produce and hold the fruit on longer and reduce the amount of drop. We are hoping to demonstrate, at least with these young groves, because these are just in the ground a little over a year now, that will also, because we're measuring tree growth, we'll see what the tree growth with and without the brass steroids and what that impact is, because the early indications are the brass steroids do help in promoting vigorous tree growth. Working with the craft working groups and the teams has been a very good experience for us. It's been a collaborative effort in how submitting the projects, working with Brandon and other members of the team and researchers and going back and forth and really honing in the projects so that it can provide the best, most benefit and the most usefulness of the information that we're able to get out of it.
Good deal. Thank you to Ron and Tamiami Citrus for assisting us with that. Tamara, the volume was loud enough, but just barely. I know you're working on it, and if there's any way you could increase the volume, that would be good. We did a dry run on all this yesterday, and things were working properly, but you know how computers are. Are there any questions for Ron? I know he's on the line.
None have come in through the chat.
Okay. Well, good deal. Well, I'm sure he's available as well. So if you have any, contact him personally, and I'm sure he'll help you in any way. Thank you, Ron, very much for helping us out. Our next presenter is Dr. Manjul Dutt, who is one of the great UF IFAS scientists at the Citrus Research and Educational Center in Lake Alfred. He works in a wide variety of disciplines, including genetics, and he oversees the citrus tissue transformation labs. He is also quick to assist CRDF on special projects, which is how this presentation came about. One of our board members, John Updike, noticed that his block of Parson Browns was doing better than anything else in the neighborhood. And so we began checking around, and that was not unique. So we contacted Dr. Dutt to ask if he could help us figure out why. That is what he is going to talk.
It appears that we lost Rick. So Dr. Dutt, if you want to go ahead with your presentation, we'll let Rick jump back in when he gets reconnected.
All right, thank you. I'm going to share my screen. Good morning. As Rick said in his introduction, I'm from the Citrus Research and Education Center in Lake Alfred, and I'm very happy to be presenting to you some of our preliminary data looking into these early maturing Parson Brown sweet oranges. So moving on, let's talk a little bit on the history of the Parson Browns. I mean, most of you are aware of it, but there are some interesting quirks to this cultivar that many of you may not be aware of. So this was identified in 1875 by J.L. Carney and named as the Carney Parson Brown. By the mid-1920s, this Parson Brown cultivar had become a very popular early season sweet orange. However, in the 60s and 70s, Parson Brown fell out of favor. And there are many, many reasons for this happening. I've summarized a few of these. Seediness, Parson Browns are seedy. It has a high relative peel oil content compared to the other industry standard at that time, Hamlin. The yields were much lower than Hamlin, and it had poor fruit quality that was obtained from blocks thought to be Parson Brown, but we speculate that these blocks were possibly not derived from Parson Brown, but would being delivered to the juice plants. So these were some other seedling selections that were like the Parson Brown, but they were not exactly the Carne Parson Brown that was identified in 1875. Since then, in the 1950s and 60s, there were approximately 15 different clones of the Parson Brown registered with DPI. And so, we can understand that at some point of time back then, Parson Brown was really important for Florida citriculture. Most of these clones were dropped eventually due to lack of interest primarily. And at this point of time, there are 3 clones that currently remain active in DPI. The first is the F56-2 that is been planted around the state. And the second are 2 relative new introductions that were introduced by Gary England here. I've put his photograph in front of a beautiful 56-2 tree. And these are the Carne Orange 229-2 and the 229-3 that he collected. And we are going to have an article on the dedicated to the Parson Browns in the October issue of the Citrus Industry magazine. And all this, the history and the introduction that I've briefly touched here for lack of time, will be given in detail in that issue. So I would ask you to kindly look at that issue if you are more interested in the Parson Browns. So this is a photograph of a Parson Brown block. As you can see, there's a huge difference between the Parson Browns and the Hamlin sweet oranges. The Parson Browns are much more healthier and than the Hamlins. As Rick mentioned, I was talking with John Updike, and he mentioned that some of these Parson Brown blocks, his Parson Brown blocks, for example, were more tolerant to HLB. And this got my interest, and so I decided to investigate, what could be some of the reasons for the enhanced HLB tolerance in the Parson Browns compared to the Hamlin sweet oranges. As we are aware, to ensure the sustainability of the juice plants, we need to provide more early season sweet oranges to the juice plants. And so Parson Brown being one of the early season sweet oranges, I decided to look into this and investigate the factors that are responsible for the enhanced tolerance. So this is a brand new project. We just initiated it in January 2021, and I will be presenting to you some of our preliminary data. This project is in no way complete, but we have learned a lot in the last 6 months. The overarching goal of this project was to understand whether Parson Brown's tolerance to HLB is rootstock independent. And we also wanted to identify select clones with improved horticultural qualities. Now, as I mentioned earlier, many clones were available and most of those were dropped by DPI. We only have, the old 56-2 clone and the newer Carne clones. But can we identify some clone apart from these among the different groves that stood out and that could be reintroduced to DPI and given to the different stakeholders for their use? So having said this, there were 3 objectives that we have in this project. We wanted to determine the effect of systemic acquired resistance and enhanced tolerance to HLB. I have been working primarily on systemic acquired resistance for several years now, and we have made several interesting discoveries on this. We also wanted to monitor tree health and HLB levels. And finally, at the end of the season, we want to conduct juice analysis and evaluate quality parameters from these selected Parson Brown trees to understand if there are any differences in any between the different locations. So before I go into discussing our project, I'd like to give a brief introduction on the systemic acquired resistance phenomenon that we're investigating. To understand this, you'll have to look into how the plant defends itself from biotic attack. And there are two main ways in which there is plant defense. The first is a physical level defense, and that's through the production of barriers such as wax and trichomes that can restrict pathogen infection. The second is through a systemic plant defense mechanism or innate immunity mechanism. And this is a huge group of factors that can help the plant defend itself from from pathogens. Many different factors play a role. Ed Stover, who will be talking later on, he's working on antimicrobial peptides. Those are also some of the components. Our group is focusing only on the systemic plant defense mechanisms. So having said this, what is the systemic plant defense mechanism? So systemic plant defense mechanism, or systemic acquired resistance, this involves the generation of mobile signals upon pathogen infection. So when you have something infect the plant, the plant reacts by producing certain signals. These signals are translocated throughout the plant through the phloem. And in theory, you should have protection of the uninfected portions of the plant against secondary infection through this systemic acquired resistance mechanism. And this is a very simplistic flow chart of the process of systemic acquired resistance in a plant cell. So when you have plant infection, the infection triggers the accumulation of a compound called salicylic acid in the plant cells. This salicylic acid then binds to another protein called the NPR protein, which is present as we call, in an oligomer form. Once it binds to the NPR protein, it reduces it to a monomer. This NPR monomer travels to the nucleus and initiates a number of PR and SA responsive defense related genes that are highly upregulated. Once these genes are upregulated, they produce these PR proteins that results in the activation of plant defense. So, this is a very simplistic way. If you want me to explain to you how this works in detail, I'll be more than willing to discuss this with you. And so our objective is to look at some of these PR genes to see how they can have a role in the enhanced tolerance towards HLB. So PR1 and PR2, these two genes that we're looking at, these are not something new to us. It's not something that I have identified. It's been known. For example, Dr. Jim Graham has been working with PR1 and PR2 for a long time. And his lab found that, when you applied exogenously different systemic acquired resistance inducing chemicals to the citrus tree, you had enhanced expression of these PR1 and PR2 genes. PR1 and PR2 also has antimicrobial activities against various plant pathogenic fungi, bacteria, and oomycetes. And these have been proved without any doubt in many different plant species. Dr. Graham's lab also understood that upregulation of PR2 was correlated with the reduction of canker lesions in citrus leaves. So this is very important. We looked at transgenic expression of NPR1, and we found that once NPR1 was highly induced in citrus, it resulted in an enhanced expression of PR1 genes, which resulted in enhanced HLB tolerance in these citrus lines. So, given together all this information, we know that PR1 and PR2 plays a major role in the SAR process. So coming to our project, this CRDF funded project, we decided to investigate and look at different blocks that grew these Parson Brown trees. All the blocks that we are looking at have trees that were planted in the 80s and 90s. And these trees have survived for the last 30 years. And in many of the blocks, I'll show you some photographs, they're doing just fine. So we are looking at 3 blocks in Polk County, 2 in Highlands, 1 in St. Lucie, 1 in Glades, and 1 in Collier. So it has a good, well-distributed distribution throughout all the major citrus growing locations. As I had mentioned earlier, we are also interested in understanding the rootstock effect. So when we look at the rootstocks, we found that half of the blocks that we are looking at had swingle as a rootstock, but we also have blocks that have Carrizo and Clio. Most of the blocks have the Parson Brown 1, 2, 3 clone, which is a early maturing clone, but the acid levels do tend to drop off quickly in these trees. And so they are no longer being planted widely, and DBI does not have a clean accession of this clone. We also are looking at the Camp Mac Grove in Likes that has the Parson Brown 1, 2, 3 and the 56-2 trees. Unfortunately, we were unable to identify some of the clones in half of the sites that we are sampling. And again, this has to do with records and the fact that these trees were planted almost 30 years back. So this is a photograph of a beautiful Parson Brown row. And I believe this is at the Premier Citruses site in Florida. And you can see how beautiful these trees look like. I mean, the photographs that I've taken does not do justice to what I'm explaining. You know, I mean, lovely, lovely trees. When you see these trees, and then you go over to the next block and you see Hamlin trees, and these are on the same, you know, fertilization, nutrition, and, you know, other regimes. And then you can understand that, oh yeah, Parson Browns does have something that's inducing it to be more tolerant to HLB. We collect fully mature expanded leaves for all our investigations, and so that we don't have any sampling bias during our collections. When we looked at the HLB levels in in all our blocks, it was interesting to understand that there was no statistical difference between the HLB, between the CLAS levels in any of the blocks between the Parson Browns and the Hamlins. So the Parson Brown trees were as infected as the Hamlin trees in all the sites that we sampled. So even though these trees are infected with CLAS, they seem to be doing just fine. And that's one of the objectives of this project, is to understand, you know, what's going on with it. When you look at the PR1 gene expression from samples that we collected in February, March, you'll see that all locations except the Immokalee location, which is at the far south, have enhanced PR1 expression. So this tells us that there is a systemic immune response going on in the Parson Brown trees compared to the Hamlins. And it is possible that this systemic immune response is playing a role in the perceived enhanced tolerance to HLB. PR2 expression was a little different. The only 2 sites that has statistically significant differences was the McKenna site in Sebring and the Like site at in Lake Wales. The other sites for the February-March sampling did not show any statistically significant results. But you have to understand, this is just one sampling. This is a brand new project that we have. And we have sampled again in June, July. We are crunching those numbers right now. We will again be sampling in September, October, and finally just before harvest. So hopefully we should be able to come up with a comprehensive trend throughout the year. So the key findings, and most of these are visual findings as we go to all the blocks. I have presented data for 8 blocks. We have looked at several other blocks that we have not collected samples. But in general, all the Parson Brown trees, irrespective of the location, have much lower fruit drop compared to Hamlin. Blotchy mottle on the leaves of the Parson Brown trees were less pronounced than Hamlin, even though trees had comparable CLAS titer. And this is something that we will investigate, because this is very interesting to me, why this is going on. And the third important aspect is that all Parson Brown trees had much better canopy density with lower dieback than Hamlin. irrespective. You go to any of the sites, you're going to see all these three trends to be consistent everywhere. So, this is an ongoing project. We are collecting, DNA and RNA for analysis. We are going to look at starch content, chlorophyll fluorescence levels, and leaf nutrients will be identified and analyzed in September. We're also going to run the fruits through the CREC pilot plant, and we'll be getting the juice characteristics. We are more interested in the percent juice and the fruit weight, and of course the pound solids. That's the bottom line for the industry. We will want to understand what the pound solids are of these different Parson Brown trees compared to the Hamlins that we collect from the same block. So, having said this, what can we do with the knowledge that we have gained from the study? The juice quality standards have changed a bit with the advent of HLB. The standards were different. And so I think it would be prudent to reintroduce the Parson Brown 1, 2, 3 and other clones that we identify into the DPI's parent tree program, get them cleaned up and make them available to any stakeholder that wants to use them. We also would like to establish larger field trials, replicated field trials of the selected Parson Brown clones on different rootstocks. When these trees were planted in the 70s, 80s, and 90s, we didn't have many rootstocks to choose from. And you can see most of the trees are on Swingle, Carrizo, and on Clio. But now with the newer rootstocks that are coming out from the University of Florida's and the USDA's breeding program, we have a much larger selection. I'm a plant breeder, and I would also like to understand the genetic makeup of these select Parson Brown clones. Newer DNA sequencing techniques have made sequencing genomes and understanding and characterizing these genomes much cheaper than they were 5 years back. And so we would like to understand what's going on with these genomes, with the Parson Brown clones, compared to the standard Valencia that is available in the database. The other practical aspect of this project would be to reduce the number of seeds and the peel oil content through irradiation, and because that is the fastest way in which we can produce a modified plant, keeping most of the other traits consistent. And although this was not proposed in the CRDF project, I have taken on this as a side project per se, and we have gotten several hundred buds of both the F56-2 and the Carne Orange 229-2, and we have irradiated them in Gainesville and in our main campus. And these buds have already been budded onto US 942, and hopefully we will be able to get this out planted in the next few months. And hopefully we should be able to collect data from these trees in the coming years. Having said this, I'd like to thank everybody who has helped us in this project. I cannot give enough thanks to John Updike, who actually helped me to initiate this project, and Gary England, who has been extremely helpful, and Brandon Page too. The other growers that I thank, have allowed us to look into their groves. Pat and Marty McKenna has been wonderful. Holly Chamberlain and her team, David Wheeler and Anthony Pasture, Glenn Begg, John Ghosh, and finally, Tommy Thayer and Greg Taylor from Southern Citrus. I'd like to thank each and every one of you for your support for this project. And I hope to collaborate with you down the road on this. And I'll also finally thank you to CRDF, for funding this project.
You bet, Manjul, and thank you very much. This is fascinating. Are there questions of Dr. Dutt?
None have come in at this point, but if any do, I will make sure to forward them to Dr. Dutt following the presentations.
Absolutely, thank you.
Thank you again, Manjul. Wouldn't it be ironic if one of the oldest cultivars turns out to be part of the solution when we can finally get HLB in the rearview mirror? But thank you again, Dr. Dutt. That was really interesting. Our next craft project involves Graves Brothers Company, a name that is synonymous with Florida citrus, especially in the Indian River area. In fact, Graves Brothers is the oldest company of continuous existence that grows Indian River citrus fruit. The company is still owned by the same family, currently being managed by the fourth generation. Always there to lend a hand to the industry, including CRDF. The presentation will be by David Howard, the Vice President of Agricultural Operations. David is also the current president of CRDF. He will be talking about rootstocks and scions and individual productive covers. Tamara, roll the tape.
Oh, give me one moment. It's not wanting to show up as an option to share. I apologize. One moment.
While Tamara's pulling it up, I know David is listening, but he's in a grove in Immokalee and his service isn't real good, but he'll try to answer questions if you have any, if he can hear you.
Okay.
I'm David Howard. I'm the Vice President of Agricultural Operations for Graves Brothers Company in Vero Beach, Florida. The Graves Brothers Company was founded in 1895 by the brothers in Virginia, and they moved to Florida in the 20s and established a citrus, timber, and vegetable operation in the Wabasso area. Today we manage citrus, timber, and vegetables still in our locations in St. Lucie County and Hendry County, Florida. This is our Ashland property of 1,500 acres in St. Lucie County that was planted in 1985 and was primarily round oranges for processing, planted on sour orange at that time. Since then, the grove has suffered through tristeza and obviously HLB. We've replanted the area intensively, resetting and replanting solid blocks to present a mix of processed oranges now that have various rootstocks and different planting densities from the original planting. This is one of 3 craft projects that we have in St. Lucie County on this property. The examination on this particular craft project the primary examination is Valencias, and 2 different types of Valencias. We have the 114-19, which is more of an industry standard, versus the BN65, which is a newer Valencia scion variety. The secondary examination here is IPCs on half of the project, and not IPCs, a regular psyllid control program on the balance of the project. So there's 2 focuses. One is the scion and the other is the IPCs. The entire project is planted on US 942 rootstock uniformly through the 30 acres. The primary examination of the Valencia scion varieties was prompted by some interesting data that supported the BN65 being a more robust vegetative scion, as well as a more precocious bearer of Valencias at maturity. So our intent was to see if we could support those claims, those early claims, by including those 2 scion varieties in this craft project. Our secondary examination of IPCs in this trial versus non-IPCs was intended for us to help support the fact that it appears the trees come out of the IPCs after 2 years with little to no infection, and to help us understand how that might give us an advantage as the trees enter maturing and bearing period. One of the additional strategies that we've deployed outside of the craft purpose was higher density plantings here in this block as well. The original plantings in this grove in the 80s were at 120 to 150 trees per acre. This planting, we've got an 8 by 25 setting that gives us a setting of 218 trees to the acre. So we're adding to our density while utilizing the existing beds and structure of the grove that was planted in the 80s. GBC endorses craft and craft's efforts to help growers deploy strategies to improve the HLB situation, utilizing the technologies that have been provided to us by our research organizations throughout the state and through the USDA. I'd also like to thank the participants that have helped us get to this point, most notably our legislative group who helps fund the craft program, and also the people at craft, their board of directors and their technical working group. We'd also like to thank our direct contact, Brandon Page, who helped us design the trials and supported us through changes and modifications to make it more meaningful. Our two scientists that have helped us along that way as well, Dr. Shatters and Chris Vincent, have been tremendous assets to help us make sure that the trial has scientific merit.
Good. Thank you, David. Any questions of David if he can hear them? Okay, well, if you have any, feel free to call David. David, you look really good on that tape, man. You may be getting a call from a Hollywood talent scout. I don't know. You look like Tom Selleck without the mustache. Excellent job. Thank you. Our next presentation is by Dr. Ed Stover. Dr. Stover is a horticulturalist and geneticist that CRDF is proud to have funded over the years. He is an outstanding plant breeder, focusing primarily on scions. He is with the USDA ARS laboratory in Fort Pierce, where he is developing breeding solutions to HLB. He will be sharing with us details of a trial that I suspect you have heard about, a trial which is known as the Pantuso Top Working Trial near Zolfo Springs. It is a huge trial involving 200 varieties. Dr. Stover, thank you for being with us today, and the floor is yours.
I appreciate the invitation. It's always great to visit with my many friends in the industry. So can you see my screen?
Yes.
Okay, so as Rick said, I'll be talking about the 200 variety top working trial, and I hope it will be useful for me to explain how this is a foundation for genome-driven selection and advancement of new varieties. I'm representing a consortium, including several major brands, research organizations, and processors. And I need to thank, of course, 2020 Grows, which is hosting the trial. You've all heard about it. The top working was conducted March 2020 in a 16-acre plot near Zolfo Springs. Trees were 5-year-old Valencia and Swingle. All were HLB positive, but well managed. The goal here is to get the trees uniformly infected with HLB quickly and to grow the trees out as fast as possible so we can get the information we want. There are 200 diverse accessions with 4 replications of each in 3 tree plots. There are 12 trees of each of the 200 accessions. The accession just means the type of plant material. The type of plant material used included 80 hybrids and somoclones with commercial potential, 20 standards of diverse genotypes, and 100 accessions that capture diversity within the citrus gene pool. So what's the purpose? The idea here is to develop the tools to more rapidly identify priority selections. Citrus breeding programs have created tens of thousands of new hybrids since 2010, targeting outstanding fruit quality with an emphasis on sweet orange-like and productivity when infected with HLB. The USDA program alone has produced 37,000 new hybrids in that period of time. We all know the time course of citrus breeding is slow, and it's normally taken 15 to 20 years from seed germination to having the confidence to release the material as a new cultivar. So all the selections we have with good data at this point in time are from crosses made before 2010. And obviously we need HLB tolerant sweet oranges and grapefruits now. So we need to greatly accelerate this process. So how will the top working trial help with this? Genome-wide and genomic selection, which I'll abbreviate GWA/GS, are founded on determining the relationship between the complete genetic makeup, the genotype of an organism, and its complete set of traits, the phenotype. For multiple traits, this is so complex it can only be accomplished through computational means, developing a genomic selection model. After the model is validated, it can be used to assess the genotype data for these thousands of new and existing hybrids, predict their phenotype for the priority traits that we have identified, of course, for the purpose of our discussion, primarily sweet orange-like and HLB tolerant, and select the individuals that most closely manage these target phenotypes. For us in Florida, that means that the top 20 new hybrids can be selected out of thousands, even before the trees are field planted. They can then be subjected to juvenility-reducing practices and advanced replicated trials within 3 to 4 years of seed germination, so that much-needed cultivars could be released within 8 to 10 years of initial seed germination. This is not science fiction. Genome-wide association and genomic selection are routinely used in major crop breeding, such as soybeans and maize, corn. So what needs to be done? The successful genome-wide association genomic selection depends on good genotyping and good phenotyping for the traits of interest in a broad cross-section of relevant genetic material. And that's where these 200 accessions can come in. USDA, UF, and University of California Riverside breeders worked with processors to select the 200 that include all combinations of fruit quality traits and HLB sensitivity. And obviously, due to our compelling need for HLB tolerant sweet orange types, we have a strong emphasis on sweet orange in the pedigree. Similarity to sweet orange parental types are hybrids that include sweet orange parental types in their pedigrees. This provides the greatest discrimination, the smallest number of markets for sweet orange-like traits to help us get where we need to go as quickly and efficiently as possible. The Coca-Cola company has already paid for 25x full sequencing of all 200 accessions, so we have the genotyping in hand. Now we need the resources for thorough phenotyping and the model development, the genomic selection model. Importantly, genome-wide association genomic selection will not provide predictions for traits not tested in the training population, so the phenotyping needs to be very robust. This is a draft admixture analysis of the first 120 accessions we included in the trial, conducted by Omar Cornejo as a contractor to Coca-Cola. The individual colors here, these are the parental types that contribute to the genetics of most of our selections. And you can see that we have a wide array of proportions of different parental species in here, which is what we need to conduct a genomic selection model. So this 200 accessions is properly called the training population. And what will we phenotype? Mostly it'll be obvious stuff, and we'll do it in the field just because we're talking about 2,000 plus trees. However, fruit samples will be sent to the lab to test for things like sweet orange-like flavor and quality of flavor, as well as bitter compounds, the delayed bitterness, which makes it difficult for us to process many mandarin types, for example, and makes navels a poor choice for processing, which you can't measure in the field. It's got to be measured in the lab, as well as aroma and flavor volatiles. Importantly, fruit will be available for processing trials and packing trials on selected accessions with our industry partners. However, this Zolfo Springs top work trial is just the start. As I say, the Coca-Cola company has funded the trial and sequencing for the training population, as well as initial LiDAR drone phenotyping to get tree growth and health measurements, which we used in blocking the initial trial. PepsiCo has just completed top work working with a subset of the initial 200 accessions at a Southern Garden citrus site, which will need phenotyping to help validate the models. And in independent trials, Coke, PepsiCo, and Florida's Natural are all completing creation of new nursery trees using a subset of the initial 200. And they'll be planted each in 2 diverse locations. And they'll need to be phenotyped to validate that the location of the training population and the top working, which of course is not a usual practice for Florida citrus, did not provide spurious associations. However, phenotyping will not need to be as extensive in these model refinement trials. Finally, the important thing, we're not just doing this for a genetic study, some academic study. Importantly, we want to apply the genome-wide association genomic selection models to selection and advancement of hybrids. Importantly, the drop-in replacements for early mid-oranges, Hamlin mid-sweet pineapple primarily, and Valencia are not in the top work trial because we don't have them yet. Maybe we'll be surprised, but I'm not expecting to find a drop-in for sweet oranges in 200 accessions. However, these will be identified or developed through the genome-wide association genomic selection in existing hybrid populations. I'm confident we have something that will be very close to a perfect, let's just say a very good replacement for sweet orange in our 37,000 new hybrids. This will also be used in helping select parents for subsequent generations of breeding programs. So these models will accelerate finding the nearly perfect selections among the thousands of new hybrids. We're going to start doing the phenotyping and selected hybrid populations of this year, because we don't need to have the model in place to do the phenotyping. And then once the model is in place, we can genotype them and use the model to predict which ones are our best selections, and also use the data we've collected to validate and adjust the models. So once we have the model in place, its prediction of target trait hybrids will give us the confidence to expedite advanced testing, ultimately making those selections as is done in corn and soybeans, for example, even before cropping, providing even better sweet orange-like selections in subsequent years. And as I say, it will also be used to select parental combinations. And at least among the breeders, we've agreed to allow our breeding material to be used for other breeding programs when they're identified as being the best choices for crosses. So ultimately, these will bring us to close to the perfect selection that will resolve our HLB woes. Just to reinforce, current citrus breeding, 15 to 20 years to release. Citrus breeding using validated genome-wide association, genomic selection, 9 to 10 years to release. You may have heard gripes that the planting is flawed. Top working is not a typical practice for us in Florida citrus. It was about 80% successful in the first effort, which pleased us. Most of the standards, hybrids, and accessions developed extremely well. Others did not, and were rebudded on the same trees, but the success rate was low in rebudding. So we've had new trees assigned and been rebudded. So if you visit the site, you'll see some dead stumps. We've moved on from those. We also need to remember that many accessions will show HLB symptoms very quickly. This is what we expected. The use of 3-tree experimental units provides us a very robust design, so the statistical power is unaffected if only a single tree is well established in each group, each of the 4 3-tree reps. 5 of the accessions have only 3 of the 4 reps intact, still good enough for our model. And these are data from Robin Bryant, so successfully top worked over 2,000 trees, failed in almost 500, about 500 retop worked, and the only number of 3-tree reps at the time of this assessment with no successful top work was 9. So we have 194 different accessions with at least 1 successful tree in all 4 reps. I'm going to show you a little bit of data about an adjoining trial, because it includes many of the same, or many of the trees in this trial of 30 are in the 200 accession trials. Collaboration between the same folks. This is a trial that was established by Greg McCullum under MAC, and so it includes University of Florida, University of California, and USDA collaborators. Just looking at biomass as measured by LIDAR, the top selections so far include Sugar Bell and Sundragon, as well as this one mandarin hybrid of ours, which is a really nice mandarin, but of course not a substitute for sweet orange. And at least so far in the top quality group is one of our sweet orange-like hybrids that we've released from the USDA program. However, it's not performed better than Hamlin in most trials than I expect, although it has looked good early. So my expectation is that that will drop behind. This is a picture of one of the reps of Valencia in this trial, showing thin canopies and not great growth. In contrast, this is our Sundragon in the same trial. And this is Sugar Bell, which looks good here as it does in most places, also known as LB8-9. I do need to point out again that while Sugar Bell and Sundragon look good, they're not drop-in replacements for sweet orange. So we've had a project for the last 3 years in which we've tried to identify hybrids that are HLB tolerant and can be used to supplement the orange juice stream to enhance quality and volume. And Ann Plato and her team of colleagues at USHRL have tested 50 of the most HLB tolerant selections. Only Sugar Bell and Sundragon are consistently HLB tolerant with orange-like flavor among the 50 that we evaluated. And you see that in this lower left quadrant of the principal component analysis, this represents sweet orange-like characteristics, while this represents mandarin-like characteristics. Whereas this is off-flavor bitter types, and this is excessively sour types. Again, Sundragon and Sugar Bell look good. Neither is a drop-in replacement for sweet orange, but both have potential for supplementing the juice strain. So in our analyses so far, and they both have looked good in processing trials to date. So this is using only USDA examples, why we need genome-wide association and genomic selection. Again, 37,000 hybrids from 554 unique parental combinations. We anticipate an onslaught of good selections. Obviously, since we started this 11 years ago, we already have some that are flowering and cropping, and we've made some good selections. We need to figure out how we can expedite testing of the best selections and how we can best predict future parental combinations. And the answer to both of these is these genome-based models. So this is an example of a variety we produce using old-fashioned artisanal phenotype-based parental selection. This is a new sweet orange-like hybrid. We crossed our two most HLB-tolerant varieties, Sundragon and 551-2, which is an easy peel mandarin. And we have a number of good selections. This is perhaps the most sweet orange-like. Taste very orange-like, similar to Sundragon, but with higher initial acid. Brix of 13 to 15 mid-October through mid-December with no trifoliate off taste, which is also true of Sundragon. And Ann Plateau in her initial assessment found the juice agreeable and OJ-like. We use this extensively as a breeding parent this last year since we made the selection. It's also been accepted by CRDF for advanced trials, even though we only have information on one tree today, just because it looks so sweet orange-like, and it is from the two most HLB tolerant selections. Here are some more selections that we have. 5-6-36 does not have any Pensiris in it, very orange-like hybrid, used extensively as a breeding parent, also included in the CRDF advanced trial. Here's some more 5-51-2 by Sundragon, just teasers. Another hybrid by Sundragon, the same hybrid by Sundragon again, very sweet orange-like. So old-fashioned phenotype-based breeding does work, but the best parental choices may not be obvious. And so in addition to accelerating advancement of new hybrids by identifying genotypes associated with desired phenotype, we are also going to be able to better select new parents. And this is an excellent example that some of our best HLB tolerant selections have highly HLB sensitive parents. This 163-85, this really good looking tree next to a scraggly Hamlin, is a hybrid between 137-12 and Sunburst, which are both among the very most HLB sensitive varieties that we have assessed. And the genome-wide association genomic selection will allow us to look past the phenotypes and select the ideal parents based on genotypes through computational analyses. And of course, I need to thank folks for USDA funding. I'm only expressing thanks for the funding I'm familiar with that is applied to our program. We've been fortunate to get good support from CRDF as well as New Varieties Development Management, NIFA. And the Florida Citrus Research Foundation provides our Whitmore farm. And these are the folks who've contributed to our progress in the lab greenhousing field. I think I've got time for some questions.
Are there questions of Dr. Stover?
None have come in at this point.
Okay. Well, Ed, if you have had 194 successes out of 200, I think that's quite remarkable. And 37,000 hybrids is quite a body of work. So congratulations.
Well, we're doing it to try to resolve the problems of the Florida citrus industry first and foremost, although we're committed to helping all of the citrus industries in the U.S. Obviously, the problem of HLB is an 800-pound gorilla that commands almost all of our attention. Thanks for giving me the opportunity to speak with you all.
You bet, and thanks for being with us today.
Hold on just a moment, Rick, if I may. Dr. Stover, a question just came in. The question was,
okay.
Once a model is developed, is it site specific?
It should not be site specific, and one of the reasons why we or the major reason why we're putting plantings in different places is to validate that the basic fruit quality and growth traits, and of course HLB tolerance, are not specific to a single site or a rootstock. And so that all has to be validated, although our expectation is that they will be pretty much similar across all sites and rootstocks.
Thank you.
Okay, good. Thank you again, Ed. Our next craft project is by Cornelius Caldwell. Cornelius is a 6th generation citrus grower. He's a person who I have had the chance to get to know over the last 8 years, and I've become a big admirer. Most of his groves are in Lake County near Tavares, and you know how rapidly that area is developing and how quickly population growth is swallowing up many of the parcels in that area. Yet Cornelius and his family continues to carry on in the face of that, and is an outstanding grower. He has submitted a compost trial for craft participation, which was accepted. And that's what you're going to hear about today. I found it quite interesting, and I'm sure you will too. Tamara, you may roll the tape.
Hey, it's Caldwell from Caldwell Citrus Groves Management LLC, Leesburg, Florida. My family's been growing citrus for about 5 to 6 generations here in Lake County. We're up to production now to about probably 568 acres. Okay, for several generations we haven't been faced with anything like greening. In the past we've had hurricanes, we've had freezes, but nothing like the greening disease. So what we have to do in the future here to combat the greening is actually change our practices, our grove care practices. And what we've done, we've looked into doing several trials with the University of Florida through the CRAFT program to start out with new plantings, to use compost, to use beneficial predators, and to use beneficial practices to combat the greening. Okay, this is our 40-acre block located up in Tavares, Florida. We're going to be planting Vernia on X639 and 942. We are using the compost trial because of its beneficial ways to combat the greening, especially it enhances the tree health. It's very competitive to grow a young tree from the stage of a young tree to a grown tree. And so what we're trying to do right now is combat the greening with compost to slow down the bacteria transfer of the greening bacteria.
Known the Caldwell family for about 15 years, and I've been working with them on this project, the CRAFT project, to try to help them make some improvement in their groves. Today we're putting out the compost at 4 tons per treated acre, and in hopes of putting some more organic matter and carbon in the upper soil profile, upper 3 inches soil profile for retained nutrients and moisture. And we'll be disking that in here shortly.
Key things that we figured out over the years of combating the greening disease is really root health and protecting the roots, making the roots more healthy. And with these trials shown us over the years that compost, imidacloprid, and other beneficial things that we're doing through their IFAS's recommendation that we can combat the greening.
Thank you, Cornelius. I don't know if he's on the line with us today. If you have any questions, if you'll get them to Tamara, we'll be sure and get them to Cornelius, and I'm sure that he will be quick to respond. But as I said, I have grown to be a big admirer of Cornelius, and he's been helpful to me in understanding some of the frustrations that many growers experience. So thank you, Cornelius, for your participation today. We appreciate it very much. Our next presentation is by Dr. Uta Albrecht, who is a plant physiologist with UF-IFAS, who works out of the Southwest Florida Research and Education Center in Immokalee. She joined SwiftRec in 2016, but before that, she was a longtime member of the citrus improvement team at the USDA ARS laboratory in Fort Pierce. She is connected to a wide array of research projects, and there seems to always be a strong extension component to everything she works on. So she is helping us find practical solutions to some of the challenges that we face. Her topic today is a good example of that. She is working on answering a question I hear from growers occasionally, which is there a difference between trees propagated by seed, tissue culture, or cuttings? So with that, Dr. Albrecht, you're recognized.
Thank you, Rick, and good morning, everyone. Can you see my full screen?
Yes.
Very good. Okay, thank you for the invitation and the introduction. And I want to start by acknowledging and thanking CRDF for funding this project now for, it's in the third year, but the project was jump-started by the UF-IFAS Citrus Research Initiative. As you all know, in Florida, citrus rootstocks have been mainly propagated by seed. Seed propagation is easy, it's very convenient, and it has worked fairly easily because in most citrus, we have the phenomenon of nucella embryony or polyembryony, which means that nucella embryos develop from the tissue, from the maternal tissue, and they produce genetically identical or true to type seedlings. In the past, rootstocks were, before a rootstock was released, rootstocks were selected for this specific trait of nucella embryony. And as Dr. Stover mentioned, the breeding process is very long. It takes 15 to 20 years. And with the HLB crisis, there is a need for releasing varieties earlier. And sometimes, as it happened in the past, for some of the USDA rootstock releases, they were released before seed trees were available to do these analysis for Nucella polyembryony. And as was recently found, 4 of these newly released rootstocks cannot be propagated by seed because they do not produce any Nucella embryos. So if these rootstocks turn out to be superior and are desired by the growers, we have to propagate them by other methods. And these methods are either cuttings, mostly we use stem cuttings, and tissue culture. Both of these methods also produce genetically identical plants, and tissue culture is the major propagation tool for many fruit and nut tree rootstocks. Just in citrus, it has not, at least not here in Florida, it has not gained in recognition. And tissue culture is also advantageous because it allows the high throughput of plant production. Another advantage of tissue culture propagation is that we don't have to rely on the seeds or on the season, and we can produce rootstocks year round, and the plants are usually very uniform. So another reason for having to rely on alternative propagation methods is the case of US 942. As you all know, in the last 2 production seasons, US 942 was the most propagated rootstock. Out of nearly 4 million propagations, nearly 1 million were just alone for this rootstock. And this led to some problems in supply because we just did not have enough seeds available. So in order to supply all these rootstocks, they had to be propagated by other methods. And DPI now includes the number of tissue culture and cuttings propagations in the annual report. And you can see here in the last production season, 20% of all propagations were by tissue culture. And the top rootstock was, of course, US 942. And for the rooted cuttings, which were only 6% of all propagations, the US 942 was also the most propagated rootstock. So clearly there is a need for other propagation methods than seed. Of course, there are many concerns. The main concern is, of course, the higher cost associated with these propagations. And some nursery owners have reported difficulties in the nursery when growing the liners and the grafted trees. And then, of course, there is the concern that they will have an inferior root system. And especially in Florida, where we are threatened by the hurricanes every year, will they stand up to wind-induced uprooting? So several years ago, we initiated some studies to resolve these issues, and we evaluated the trees in the shorter term through the different nursery stages, and also in the longer term, and these studies are still ongoing, to evaluate the root structure, the field survival, and field performance during the early years after planting, and hopefully also throughout the productive years. So, and in all our trials, we include different rootstocks just to make sure there are no interaction effects of a rootstock cultivar and the propagation method. So I want to start a little bit by summarizing some of the observations during the nursery stage, because that's important to understand the root structures. So when we grow these different rootstocks from seed, cuttings, or tissue culture, we see differences. When we grow a rootstock from seed, plants usually will develop one well-defined single taproot with some lateral roots adhering from it. When we propagate them by cuttings or tissue culture, instead of one single taproot, we have an adventitious root system where multiple roots originate from the base of the plant. And these multiple adventitious roots in turn have fine lateral roots. So the root systems are very different. What does that mean for the early liner stage? We have done lots of studies and they're all published if you need the details. So we have a ton of data. I just want to summarize all these experiments. What we found, not surprisingly, was that the seed propagated rootstocks produced mostly one well-defined taproot, but that tissue culture plants and cuttings produced 4 to 8 adventitious roots, with the cuttings usually more in the higher range. And one observation that is maybe validates some of the nursery owners' concerns is that the tissue culture and cuttings plants had a smaller root to shoot ratio and more finer roots than seedlings. There's nothing bad about that. It just means that you have to watch out that not to over irrigate or over fertilize these plants. So let's take a look at the field-ready grafted trees. So we observed the plants throughout the grafting stage, and then before we planted them in the field, we took a detailed look. And here you can see how the root structures look before planting. And these trees were all produced in a commercial nursery, so they were not handmade by us or anything. So all these trees underwent the same process. So here on the left, you can see a seed propagated rootstock, and then cuttings, tissue, and different tissue culture rootstocks. And you can see what we also saw in the liner stage is still evident, the different root architectures, the single taproot on the left, and the multiple adventitious roots for the other propagation methods. But I want you to take a look at the arrows here. What you can see is that no matter if we have a seed propagated or otherwise propagated roots plant, we can see that once the main root reaches the bottom of the nursery container, they will stop growing. And just keep that in mind for later during the seminar. So the take-home message for this part of our study was that the differences in the root rates persisted throughout the grafted tree stage. And we also saw still some differences in the root to shoot ratio, but the differences were not as evident as during the liner stage. And we also noticed that the root growth was restricted by the dimensions of the nursery container. So of course, this is all irrelevant unless we test it in the field. And for this purpose, we have several field trials in place. 3 of them I want to talk about today. We have a couple other ones going on. So the first trial was a trial at SwiftRec here in Southwest Florida at our Research and Education Center. It went for 2 years, from 2017 to 2019. And 2 other trials are in with commercial growers. One is with Duda Ranches in Hendry County here in Southwest Florida, and the other one, Peace River Packing Company in Polk County on the Central Ridge. So the nice thing about these 2 trials is they're in a different environment, not only because the soil type is so different, also different production environment. And as I said, the trees in these trials were produced in the same nursery, so we just basically split them up when we planted them. So let's take a look at our SwiftRec field trial here. And in this trial, we compared seed, tissue culture, and cuttings. And the scion was Valencia, the standard 11419 clone. We used 6 rootstocks, Swingle, 802, 812, 897, 942, and 1516. So what do we do when we want to look at the roots of our tree? So the only thing we can do is really to dig. And how do we do that without destroying the fibrous roots? So what we did, we bought an air spade, and you can see that here is a big nozzle. It's called the supersonic nozzle, and it produces highly compressed air. And I'll show just a quick video here to see how we did that. So you can see it's a very messy undertaking, but we were successful in excavating 72 trees in a time frame of about 3 weeks. And you can see how the trees look here. The root system looks a little bit sad because of all the dirt, but nevertheless, the fibrous roots are all intact. So for us to do a detailed analysis of this. So what we did, we looked in detail at the root architectures, and we measured the root crown depth. Then we looked at the main structural roots, how many there were, and how thick they were at the base of the rootstock trunk. We also measured the length of these main structural roots, and we counted the number of lateral roots and the total root length. And you can see the roots look very black here. That is not because they're sick or dead. That is just because of the dark soil we have here, but they're very healthy otherwise. And here you see a table with some of the data where we compare seed, cuttings, and tissue culture, abbreviated here with SD, CT, and TC. And as I said, we measured the root crown depth, the total number of main structural roots, the total length, the diameter, the average length, and the number of side branches. And here with the red bar, see the statistical results. When I have an NS that says that means they are not statistically significant, any differences. And when I have an asterisk, that means they are statistically significant, the differences. And the more asterisks there are, the higher the significance. So as you can see, for most of these root rates measured, we did not find any influence of the propagation method. The only difference we found was the average length of these main structural roots, which was a little bit higher in the seed and cuttings propagated rootstock in this trial. So let's take a look at these root crowns again. Here you see 2 root crowns from seed propagated rootstocks. And as you maybe notice, never ever have we seen a single taproot in any of the trees that we excavated. So you may think it's Southwest Florida, we have higher water tables, but I argue that one of the reasons for this is just simply that we're using a nursery, that the nurseries now grow the trees in pots as opposed to be outdoors. So as soon as the roots hit an obstacle, they will stop growing and then turn into a different direction. So let's look at the root architectures of the different propagated rootstocks. You can see seed, cuttings, and tissue culture root crowns. And you can see there's really hardly any difference. Again, no taproot. The roots just don't go very deep, and you couldn't really tell them apart. So let's take a look at the total tree biomass. As I said, we excavated the whole tree, we took them apart, we separated all the tissue, and we tried it and weighed everything. It was a lot of work, but we got some good data. And as you can see here, there was no statistically significant difference on the whole tree biomass, no matter how the rootstocks were propagated. When we look at the different rootstocks, however, we did find strong, significant differences in total tree biomass, with US 812 and 942 having the highest biomass, and 897, not surprising, and also 1516 with the lowest total tree biomass. We also looked at the biomass distribution. We separated the trees into the roots, the stems, and the leaves. And you can see, again, no significant differences based on the propagation method, but we did find significant differences among the rootstock cultivars, but I'm not showing the data here because of the time. And we also looked in detail at the root mass fractions. So for this, we separated the whole root system into the root crown, the lateral roots, and the fibrous roots, and we tried it and weighed it. And again, as you can see, there was no significant difference, no matter how the rootstocks were propagated. But we did find significant differences among rootstock cultivars. So the take-home messages from this 2-year field study was that there was little influence of the propagation method on the root architectures after the 2 years that we grew them in the field here in Southwest Florida. And this was somewhat different than what we observed in the nursery. So it seems the trees outgrow their differences. And we also found that the tree growth and the root architectural traits were influenced more by the rootstock cultivar and also by other factors. So for these other factors, I mean, for example, the nursery container and also the bed geometry. I haven't shown these data, but we also looked at that, and we found that more roots are growing towards the bed side and less towards the furrow side, and that the roots that are growing towards the bed sides are also longer and thinner. Maybe that's not all that surprising, but I just wanted to mention that. So here is a photograph from one of our commercial field trials in Hendry County that has been going on since April 2018 with Duda. And for this trial, we're comparing 4 different propagation methods, so seed and cuttings. And you can see here TC1 and TC2. That means we got tissue culture propagated rootstocks from 2 different nurseries. So they each use their own tissue culture propagation method. And we thought it would be good to try that out too, if there are differences among those different tissue cultures. Scion again was our standard Valencia scion. We have 4 rootstocks in this trial, 802, 812, 897, and 942. There are other rootstock propagation method combinations in this field trial, but these were the only 4 rootstocks for which we had all 4 complete propagation methods for a complete factorial experimental design. Here's the Polk County trial. Again, this were from the same nursery production going on since April 2018. This, if you recognize it, this is actually the cover photograph from this month's Citrus Industry Magazine article. So here we compare the 4 different propagation methods too, and Scion Valencia again. And here we only had 3 rootstocks, 802, 812, and 942 for a full factorial comparison, but we have other rootstocks in this trial. So some of the data that we collected in the 3 years that these trials have been going on, you can see we measured tree height, canopy volume, scion and rootstock trunk diameter in all these 3 years since the project has been funded by CRDF. The top half of the table shows the differences among propagation methods. And here you can see that for the tree height, we found no significant difference among differently propagated rootstocks. And neither did we find for the scion trunk diameter in 2021. For the canopy volume and for the rootstock trunk diameter, though, we found some statistical differences, which indicated that in 2021, the seed propagated rootstocks produced a little bit of a higher canopy volume, and the rootstock trunk diameter was also larger, but not by much. When we look at the comparison among rootstock cultivars, we can see canopy volume was not significantly affected, but most of the other variables were. For example, the tree height was largest for US 802. Also, the rootstock trunk diameter was largest for this rootstock, which is no great surprise because US 802 is known for its vigor-inducing effect. So let's take a look at the data for the Polk County trial. Again, we measured the same variables. And you can see here, which was very interesting, that even though the trees were produced in the same manner, were planted at the same time, we did not find any significant difference in this trial based on the propagation method in any of the years. We did, however, find significant differences for the rootstock cultivar. And here again, like for the Hendry County trial, US 802 had the largest values for all the traits measured. But the tree size is not really as relevant as the yield and the fruit quality. the trees are only 3 years old so far. So this year we had the first somewhat yield. So we collected all the fruit. You can see on the left here the Henry County trial, the table in blue. The average yield was not very large yet because the trees are there still on a young tree management program, but we measured it anyway, and you can see there was no difference based on the rootstock propagation method, but we did find differences for the rootstock cultivar and with US 942 producing the largest yield per tree. We take a look at the Polk County trial, the table here in green. We can see that for the propagation methods, again, we did not find any significant differences. And in this trial, the yield was on average about 3 times as high as in the other trial. So we also were able to measure the fruit quality, but also there was no significant difference. When we look at the rootstock cultivar, though, we found some significant differences, but maybe it's not that relevant at this point because it's just very early for fruit quality and it's not expected to be that great. So to conclude, there was no major influence of the rootstock propagation method on tree growth during the early years in the field. And the influence of rootstock cultivar on the tree growth was larger. And there are certainly other factors that influence the tree performance. And by other factors, I mean where the trees are growing. Do they grow on the central ridge? Do they grow in Southwest Florida on the heavier soils? And the tree management, and also probably other factors such as different disease pressures, climatic conditions, etc. So at this time, there does not seem to be any reason for concern when using rootstocks that are propagated by other methods than by seed. But hopefully we can continue with these studies for 3 more years at least to get some more data on the yield and other factors of tree growth in these trials. And before I conclude, I want to acknowledge all the collaborators on these studies so far: Dr. Kim Bowman, Acromilora, Phil Rock's Nursery, Brightleaf Nursery, and Duda Ranches and Peace River Packing Company. And lastly, I want to give a shout out to my awesome team here at SwiftRec, who never complain, even if they have to dig up trees in the field, and who are just, without who I couldn't do this. Thank you.
That was fantastic, Dr. Albrecht. Thank you very much. Do we have questions for Uda?
We do have one. The question is, can this rootstock analysis methodology be used to evaluate performance under different compost and ground cover practices?
Yeah, of course they can. Yeah, it's just when we're in commercial production and we want to excavate the trees, there are some other things to consider. Do we want to destroy the trees or not? So. But yeah, you can. I have actually field trials going on with Dr. Strauss. One compost trial that's currently ongoing, also CRDF funded, where we're not going to dig up the tree, but we're doing all the other evaluations. We're also looking at some fibrous root traits that I didn't talk about today. We're regularly digging up roots and doing some physiological and other measurements to see if there are differences. Dr. Strauss does the soil microbiome analysis. So yeah, certainly we can do all these things. And we also, I didn't mention it, we have a lot of data where we're using mini rhizotrons. These are these plastic tubes. I think Dr. Strauss had one in her photographs. We have these going on in these commercial trials with Duda and Peace River. I just didn't show the data yet. So we're collecting, I think every 2 months in these trials, we're collecting root growth data. So far we don't see any differences among propagation methods, but we're getting some really nice data on when the roots are actually growing. And we also have a field trial going on right now with the Bobadere and at the East Coast in Indian River site. And we are currently analyzing those data on root growth too. And we're going to do at the end of this year some uprooting analysis where we're using a winch and a force meter, and we'll try to measure if the roots are more resistant, the root systems, how they hold up to different pressures to simulate like hurricane force winds. So there's a lot still going on, but with the 20 minutes, I can only present so much.
Well, you did great, and that is very helpful and practical advice for growers. It's a project that has a hard deliverable, and thank you very much. Other questions, Tamara?
None that have come in at this point.
Okay.
Thank
Well,
you.
Dr. Albrecht is in Immokalee. If you have questions for her, I'm sure she'll be quick to respond. Thank you again, Uta. One of the great names in American agriculture is A. Duda and Sons. One of the reasons for this is because it is such a great American success story. From the first 40 acres purchased by Andrew Duda in the early 1900s, the company grew to over 45,000 acres of owned and leased land across the United States. This fifth generation family-owned company has a stated mission to multiply and share God's blessings, and states clearly the core values that drive its business dealings. What great people and what a great company. Joby Sherrod is an active and outstanding participant in helping CRDF evaluate research proposals. He is also the director of Grove Operations for Duda. He will be speaking with us about a craft rootstock and scion trial. It is replicated and the scientific design is outstanding. I think you will find it quite interesting. Tamara, roll the tape.
I'm Joby Sherrod, Director of Grove Operations here at A. Duda & Sons in Felda, Florida. A. Duda & Sons is a family-owned agribusiness. It's been in business for 96 years. We are located in the LaBelle Grove, or the Felda Grove, and one of the blocks that we have the craft trial planted in for this year's experiment. This project fell under the direction of our project leader, Brandon Page. Working with Brandon this year has been real easy. We've worked with Brandon in the past on many other projects as well, and it was very easy to work with him and establish the design, establish the statistical outcomes that we were looking to get from the data that we're going to collect. And ultimately, I think it resulted in a very nice trial that we expect to see some real positive results from. So this year we decided to look at a scion and rootstock comparison trial of a new variety that, or at least it's new to us, a variety called Salustiana. And we're comparing it to a standard of Vernia. So there's 2 scions and 7 different rootstocks in this trial. Salustiana is a seedless, early maturing orange with Valencia quality. It originated in Spain, and in a 2020 article written in Citrus Industry Magazine by Drs. Grosser, Gemitter, and Castle, they reported that Salustiana in a trial planting was showing some good HLB tolerance. So last year over the summer, these trees became available to us, so we decided to apply for a craft project with these trees, and we were able to plant them in December of 2020. The trees are planted in a randomized complete block design with 6 trees per plot and 8 replications of each plot. We have, like I said, 2 scions, Salustiana and Vernia, with 7 rootstocks, with a standard rootstock being US 942, comparing to several rootstock selections from California. Those are C22, C35, C54, C57, C147, and Willets. The goal is to get a productive, early maturing orange that is at least reasonably HLB tolerant here in our grove, in our conditions. The craft program is an important program for the industry with its support of new plantings, along with coupling with the experimental or the trial nature of the projects, where we get to evaluate different practices that hopefully make positive impacts to HLB. It certainly is a good incentive to growers to try some of these new projects that they may not have otherwise wanted to try.
That is a really interesting trial. I know Joby is online with us. If anyone has any questions, I'm sure he'd be willing to respond. Do you have any questions, Tamara?
I do not have any that have come in.
Okay, well, Joby will be available, I'm sure, so feel free to reach out to him if you have any questions about what he has going on there, but that is a really interesting trial. Our next presenter is Dr. Tripti Vashist. She is another one of the excellent scientists at the CREC in Lake Alfred. Her master's degree is in science and technology, and her PhD is in horticulture. Both of these degrees came from the University of Georgia, so she's a bulldog, but she's a gator now, of course. Her extension responsibilities focus on citrus production needs and implementing horticultural practices to improve citrus production, tree health, and fruit quality. Her presentation today is on good nutrition management and how it can improve HLB affected sweet oranges. Dr. Vashist, you have the floor.
Thank you, Rick. All right, so thank you for the introduction. In my today's presentation, like you said, I'll be talking about the nutrition management trial that I've been doing for 5 years now. The trial has been funded by CRDF, and initially it was a 3-year trial, and then we were able to extend it further. So the goal of this trial is to evaluate the effect of controlled release fertilizer with elevated levels of micronutrients and adjusting the soil pH at the same time. So there are a lot of things that are ongoing in this trial. And just to be clear, so we are talking about constant supply of nutrients, soil applied micronutrients, micronutrients at a higher rate, and then soil pH amendment. And before I get into the trial, I would like to show some background information why we were focusing on these 4 parameters and how it is relevant. So first of all, constant supply of nutrients. We do know that HLB trees have smaller root mass than the healthy trees. So, and in addition, we also know that in this graph, you can see that the root to shoot ratio in HLB trees is almost, is lower, significantly lower than that of in a healthy tree. So there are fewer roots that are there to support the shoot system. And this is a table taken from one of our publications where we compared different controlled release fertilizers, and we found that with the use of controlled release fertilizer, we were able to have pretty good yields in these 4-year-old Velcorius trees. So use of controlled release fertilizer definitely seems promising because it can constantly supply the nutrients to this diminished root system. Then talking about the soil applied nutrition program. So plant basically takes up the nutrients in a solution form. The nutrients, when they are dissolved in water, irrigation water. So you have fertilizer on the ground. When your irrigation turns on, there is a solution of the fertilizer. And then these nutrients are taken up by the plant. And they are distributed throughout the canopy wherever the need is. So the advantage of a soil applied nutrition program is that the mobile and the immobile, both types of nutrients are uniformly distributed within the plant. I will get back to the mobile and immobile nutrients in a couple of slides because that is really important when you're thinking about a fertilizer program. On the contrary, in a foliar nutrition program, you are applying fertilizer to the leaves. Now, one thing to consider is that citrus has a really thick cuticle, and that cuticle can limit the nutrient uptake. Significant amount of nutrients do get washed away when you apply foliar nutrients. Now, pre-HLB, our root systems were massive, and therefore our root system had a chance to take up the nutrient from the ground, even if it cannot take from the leaves. With HLB, the root system is small, so that limits how much nutrients can be taken up of these washed away nutrients. And another thing that is really important is that with foliar sprays, the immobile nutrients get locked and immobile. So now talking about the mobile and immobile nutrients. Mobile nutrients, like the name says, these are the nutrients that can move freely within the plant. So they move wherever the new growth is. To give you an example that we all have seen, like nitrogen, highly mobile nutrient. We always see nitrogen deficiency in the older leaves because it can easily go to the newer growth and support that. Nitrogen, phosphorus, potassium, magnesium, and sulfur are our mobile nutrients, and they are transported in xylem and phloem both. So foliar and soil applied, both nutritional programs would work with these nutrients. On the contrary, the immobile nutrients do not move well within the plant. They get logged wherever you apply them. So if you have applied a manganese foliar spray, it is logged wherever it was applied. So if you applied it in the spring of the year, so it will get on the spring flush, it will not move to the summer flush. And therefore their distribution is not as good in the tree. And most of the immobile nutrients happen to be the micronutrients. And traditionally we have been applying micronutrients as foliar sprays. So these sprays need to be timed with the growth. Whenever there is a new growth, you need to apply them because you want to make sure your new growth has it. Soil applied nutrients have an advantage because soil applied nutrients are transported with the stream of water. As your new growth will take up more water, the nutrients will get distributed. So with immobile nutrients, soil applied makes more sense. Moving to the higher rates of the nutrient, why we wanted to look at the higher rates of the nutrient. And with HLB affected trees, we have seen deficiency of nutrients. This has been done by Dr. Morgan. We have also recently published some work. And in these graphs, what I'm trying to show you, manganese, zinc, iron, and magnesium. The last two bars are from healthy trees and HLB trees that were not fertilized in each of these graphs. The first two bars are the trees that were well fertilized within healthy and HLB. And what you can see is that consistently in all of these, HLB plants have lower level of these nutrients than healthy. So somehow healthy trees were more forgiving, or they were not utilizing as much nutrients, but HLB trees are utilizing more of these nutrients. It could also be that with the reduction in root mass, there are just fewer roots to take up those. Physiological system is compromised, so maybe these nutrients are utilized in that. And we do know a lot of these micronutrients act in plant defense response. So maybe they have some role, that's why they're used more. In the same work, we had seen when we applied the fertilizer, a lot of these pathways that are related to pathogen and pest attack were upregulated upon the availability of the nutrient. So HLB trees do respond to these nutrients, and they respond to the higher levels of nutrients. We have been seeing that in other work. And lastly, talking about the soil pH, that is something that we have talked a lot in last few years. Soil pH is really important because it kind of influences the availability of the nutrient that are in the soil. In low pH conditions, your micronutrients are bound to the soil, and they are not, in low pH condition, your micronutrients are more available. In high pH, they are bound. We had done this study where we found when we irrigated the plants with 5.8 irrigation water, 7 irrigation water, and 8 irrigation water, HLB trees showed a rapid decline. About 80% of the leaves dropped and about 40% of the plants died. So only HLB trees are responding to that high pH in this way, in a worse way than the healthy trees. So this is the picture. You can see within 60 days of irrigation with pH 8 water, our HLB trees were pretty much dead. Healthy were not good either, but HLB were literally dead by then. And we have seen that it is from the same study. And what I'm trying to show here is that no matter what you irrigate the trees with, so in this case, we are irrigating the trees with 5.8. This is what we are irrigating with. But the trees have a tendency to change the soil pH. So even though we were watering every other day, we were not able to get to the 5.8. It was a long-term effort. And so the trees tend to change the soil pH to about 7 some through their physiological processes. So a soil application or a soil pH amendment needs to be a continuous process. With all of that information, I'll move to our micronutrient field trial that we did. This field trial was done or is being done at 2 locations in Central Ridge, in Fort Meade and Arcadia. We are using Valencia on Swingle, which were about 10 to 15 years when we started the trial back in 2016. This is a completely randomized block design. We started the trial in February of 2016, and we ended the first part of the trial with the harvest in 2019. We have been, and then in 2019, we got funded for another trial or extension of this trial. Now this trial will be for 5 years. So we will have 5 years of yield data when we end the harvest in 2022. We also added some more treatments, but in this presentation, I'll be only talking about the original trial. All the fertilizer treatments are hand applied in the wetted zone. So we actually make sure that we are targeting the wetted zone of the tree. We have split the application of the fertilizer in 3 applications. These 3 applications are in February, July, and early October. And one thing that I do want to point out that they are not equally split. We try to put out about 45% of our fertilizer in February, 35% in July, and about 20% in October. We do not want to feed trees in when they are getting lower in their activities during the winter months. So we try to put out about 75% of our fertilizer by the end of summer. These are the 10 treatments, the original trial that was started in 2016. What you will see in these trials, since there are 10, it can be a bit complicated, but our control is our treatment 1, which is the conventional or the regular dry fertilizer with foliar micronutrients. And then we have our tiger micronutrient mix to instead of the foliar. So you will see tiger micronutrient mix in all of these treatments. And from treatment 4 onwards, we are using controlled release fertilizer. And then treatment 5 onwards, you will see each treatment has a different amount of micronutrients. So whether it's manganese, zinc, it could be elevated by 50% or 20%. So because with the limited resources, we cannot do all the factorial design. We picked what seemed most reasonable at that time. Talking about the rates of the nutrient. So the base applied fertilizer is 12-4-16. And we have 5% calcium and 3% magnesium. So all the CRF, well, all the treatments 1 through 10 gets this. In case of the control treatments where we are applying the dry mixes, we are applying about 180 pounds of nitrogen per acre, and the CRF is 150 pounds per acre. So we have reduced the amount of nitrogen in the CRF treatment, and therefore in the CRF treatment, phosphorus, potassium, calcium, and magnesium are also reduced by 15% less of these nutrients. Our micronutrient mix, which is Tiger micronutrient mix, it's manganese, zinc, iron, and boron mix 6631. We are applying it at 225 pounds per acre. This is what we are applying. Manganese and zinc are about 12 pounds per acre, iron is 6 pounds, and boron is about 2 pounds per acre. When we elevate the levels by 20%, this is what we are applying. And these are based on the IFAS recommendations from the nutrition guide. Talking about the results, so first the soil pH. In these two graphs, you are seeing Fort Meade soil pH and Arcadia soil pH, and the x-axis is the time range. So we started the experiment in 2016 and until 2020 results I'm showing you. And we have all of these 10 treatments. We did see a decline in the soil pH as we used Tiger mix. That was anticipated. It is somewhat of a, it's interesting that the pH did not change for the first 2 years so much, and then all of a sudden it dropped. And that I find pretty interesting. Our control stayed pretty much constant throughout. So in 2019 fall, when we found our soil pH has dropped in lower fives, we stopped the use of Tiger mix. And that is something that we all should keep in mind, that if your pH drops at lower fives, you should stop the use of acidification. We want the pH to be 5.8 to 6.5. So in the yield, we did not see any significant differences in the yield for the first 2 years. Then in third year, we started seeing significant differences. We have not seen any changes in the canopy volume, significant changes with our treatments altogether, but in Arcadia, which is our southwest location, we are seeing treatment number 4, 5, 7, and 10, which are listed here. They are showing more canopy efficiency. So we are getting more fruit per canopy volume in these particular treatments. We are not seeing the same treatments to be significant in Fort Meade. It's only in Arcadia. And overall, when we consider Arcadia and Fort Meade together, we are seeing treatment number 4, which is the use of CRF with micronutrient mix, that is consistently high yielding at both sites. Here, I'm showing you the cumulative yield for these treatments in the 2 locations, Fort Meade and Arcadia. Treatments 1 through 10 are on the x-axis, and the yield is in boxes per acre. It's a 3-year sum. The first bar in both the graphs is our control, and then these are just numbers. So in case of Fort Meade, 9 and 10 is the treatment that is a combination of manganese and boron. The 9 is 20% increase rate, and the 10th is the 50% increase rate. In case of Arcadia, treatment number 7 has been our best performer, and that is the treatment with iron. So since I have so many treatments and 2 locations, I rank them based on the cumulative yield from these 3 years. And based on the cumulative yield, in Arcadia side, treatment number 7 has been our best performer, followed by treatment number 4. And then control is at the bottom. In Fort Meade, treatment number 9 is our best performer and control is at the bottom. The interesting part, and I've showed this many times, and this is that the best performer in Arcadia is our worst, or the second from the bottom in Fort Meade. And our best performer of Fort Meade is second from the bottom in Arcadia. So we really need to pay attention to the fertilizer program. It cannot be a copy paste from one location to another. That's what I had done in this experiment. So it needs to be more thoughtful than that. Now these are the yield from 2020. In 2020, we did still, the yield is in boxes per acre. So treatment number 7 is still on the top in Arcadia, and treatment number 9 is still on the top in Fort Meade. We are seeing some reshuffling happening with the control treatment somehow. Control treatment is number 3 in 2020. So we need to monitor it for another 2 years to make sure that this was just not an anomaly. It is still, treatment 1 is still not as good in Fort Meade. And 9 again is at the bottom in Arcadia side. So why these differences? So we looked at the soil nutrient results more closely. What were the soil results in the start of the experiment? So I'm talking about 2016. I have 2 rows. First one is Fort Meade and the second one is Arcadia. And if we look closely, Arcadia is really low in soil iron compared to Fort Meade, and this is the state average. And I know this is the state average based on the citrus nutrition box data that I have. So Arcadia has really low iron. It also has pretty low zinc. And we are seeing that these treatments are the ones that are performing better in Arcadia. And then manganese and boron, I don't see a similar trend there, but boron definitely is lower in Fort Meade site, and we are seeing better response there. So, like I said, iron has been found low in the soil and the leaves from southwest growing region in nutrition box. So this kind of explains that why iron treatment is doing better in Arcadia. Moving to the leaf results, this is also something interesting. I'm only going to show you the three nutrients: zinc, manganese, and iron. And here on the x-axis is the timeline from 2017 to 2019, and on the y-axis are the different nutrients. So in this case, first it's Fort Meade zinc and Arcadia zinc. These thick lines are our control treatments, or where we applied the foliar micronutrients. And we pretty much started about the same, but with the foliar micronutrients, we saw higher levels in the leaves, and our treatments had lower levels altogether. It kind of seems to converge eventually at the end. So what I'm trying to show, if you switch to a soil applied program, you will see these patterns. So somehow the leaf foliar nutrients are, when applied, show higher accumulation in leaves, whereas when you apply soil, because it is more distributed, it is better distributed in the root, in the trunk, in the fruit, you see lower levels. Eventually it will get to the equal number, but we don't see that in case of iron. So our foliar treatments, they show better numbers in the leaf, but our high yielding ones are here at the bottom. We did do some correlation analysis, and with the correlation analysis where we were trying to relate leaf nutrients to certain parameters, we found that the fruit size increased with the leaf nitrogen, phosphorus, potassium, magnesium, sulfur, boron, and manganese. And we also saw that the bricks, total soluble solids, increased with the leaf nitrogen, magnesium, sulfur, boron, and manganese, and with the increase in fruit size. So this was good that as the fruit size increased, we had higher bricks, not lower in this study. And we did see decrease in the acid with the same treatments. Overall, we found that the yield increased as the leaf nitrogen, magnesium increased, and higher yields were seen when we had the higher fruit size. Interestingly, we saw a decrease in the yield with leaf zinc. This is something that that is really, I don't know how to explain that, but overall we are finding that zinc is really important in HLB trees. We have multiple studies that are showing that HLB trees are metabolizing higher rates of zinc than the healthy trees. In some, we have been seeing that consistently. The plant defense response goes up. We know HLB trees do show more zinc deficiency than any other nutrient. So there is more need on studying the role of the zinc, but we are seeing that yield increased as there was a decrease in zinc. Possibly the zinc was more metabolized in producing that fruit. I cannot say that for certain. And when we looked at the consumer studies, we found that the overall liking increased when the fruit had, when the treatments had more zinc in them. And that was consistent. This was a work done by Dr. Yu Wong. And we also found that our control treatment had more of the compounds which are considered to be negatively associated with fruit liking. So treatment 1 had more of these, and treatment 6, which is zinc, had less of these, or least of those. To end my presentation, HLB affected trees do benefit from micronutrients at higher rate than the recommended what is recommended for the healthy trees. 20% higher than the recommended rate seems to be good for the HLB trees. Iron and zinc treatments are performing better in Arcadia. Manganese treatments are performing, manganese plus boron are performing better in Fort Meade. Soil applied nutrients seems to be better than foliar in long term. magnesium, sulfur, boron, manganese, and nitrogen are showing to impact the fruit quality positively. With the use of CRF, we are able to reduce the amount of the nutrients applied. And the bottom line is the goal or the focus should be on the constant supply of nutrients, and soil acidification is important. However, soil pH needs to be continuously monitored. We don't want to go below 5.8 in most of the cases. With that, I'd like to thank the funding agency CRDF, Dr. Jude Grosser, Dr. Wong, Peace River Packing, Orange Coalico, for helping with this trial, Matt Shook and Trey Whitehurst from Harold's, Jack Zahn from Tiger, and my team who has been doing all of this hard work. So thank you. I'll be happy to take any questions if we have time.
That was excellent, Tripti. Thank you very much. And we do have a number of questions. One of the questions I see, though, that comes from one of our growers is asking if you could show the control treatment slide again. So while you're going to that, Tamara, if you want to begin going through the questions,
Sure.
that would be good.
Sure. The first question is applying sulfur to lower soil pH as effective as applying acid to the water to lower both water pH and soil pH?
Sorry, I'm really sorry. I was thinking, I was looking into sharing the slides. So
I know I confused you. That was my fault, Tripti. I'm sorry. I distracted you.
Okay, I'll answer the question first. So the use of sulfur is as effective as
applying acid to the water to lower both water pH and soil pH.
Yes, in short or in very loose term, I mean, your goal is to adjust the pH of the wetted zone. You can do it by two ways. You can adjust the irrigation water pH, so the wetted zone receives the water at the correct pH. Over long term, you will see the differences. But use of sulfur is more of a long-term strategy, or you can incorporate a little bit in your fertilizer program each year. Whereas if you are adjusting it with the irrigation water, you have to do it with every irrigation cycle. So the goal is same, but there are different ways of getting to that goal.
Okay. The next question was, how is the canopy volume measured?
So we used a formula that has been published so many times. It's called a prolate spheroid method. So we measured the diameter and the volume. I did not mention, but we also used canopy density measurement, because I do think canopy density is more relevant than the volume, because we know HLB trees drop a lot of leaves. So you may still have a big canopy, but a very thin canopy. We measured canopy density. We have not been seeing any statistically significant results.
Okay. What is third year significant difference referring to? Yield or tree condition?
Yield.
Okay. And then would a fertigation program every week work as well as CRF?
We have not tested that. This was strictly a CRF, but my educated guess would be as long as you are applying nutrients constantly, you should be able to achieve similar results, if not same.
The next one was a 20% increase on micronutrients. Is it an increase of what standard?
So using what is recommended in the nutrition guide. So for example, for manganese, the guide says 5 to 10 pounds. I used 10 and then added 2 more pounds to it.
Okay. And then one more question just came in of, again, how did you measure the canopy density?
We use an instrument called Plant Canopy Analyzer. This instrument basically measures how much light is getting through the canopy and also how many leaves are there. So it measures a leaf area index along with how much light is getting through. So the idea is, if you have a dense canopy, less light will go through the canopy. If you have thin canopy, more light will go through.
And then the only other question was what Mr. Dantzler had already referred to, if someone requested that you show the control treatment slide again.
Okay. So I'm assuming they want to see what the treatment was. This is the treatment, which was regular fertilizer with foliar micronutrients. And what we were applying is, we were applying 180 pounds of nitrogen per acre. Foliar micronutrients were, again, according to the nutrition guide, they were strictly foliar sprays, split in 4 applications in the year. And I don't know if there was interest in seeing the yield. That's what you wanted to see. The first bar is the control in both the graphs.
Other questions?
And one thing that I did not mention here, the p-value that I'm using is 0.1. It's not 0.05. I'm dealing with 10 treatments. There's a lot of tree-to-tree variability. So I can just say that with a p-value of 0.1, I have 90% confidence instead of 95% confidence.
I see a question asking, how did you measure canopy density? I don't know if you've answered that yet or not. Perhaps you have.
I did. It is with an instrument called Plant Canopy Analyzer, which measures the number of leaves and the light that penetrates through the canopy.
Okay. All right. Well, that was excellent. Tripti, thank you very much for being with us today, and especially for the work that you're doing on this project. A couple of housekeeping things before we rise. One, you may receive CCA education credits for your attendance today. Also, there is a good chance that there will be a Craft 3. The legislature this past spring directed that CRDF spend $5 million on large-scale field trials. So we published an RFP. The time for us to receive proposals is almost through, and we expect to receive one from the Craft Board. Our process with the CRDF review of the RFP will be completed by the end of August. So if there is a Craft 3, and I suspect there will, you'll need to pay attention if you would like to participate in that program. But I think you'll agree with me that it's an excellent program and it's going to yield some very helpful results. The CRDF has 2 primary research committees and a board of directors. All 3 of those units are comprised almost exclusively on growers. We try very hard to fund the projects that we think will help you in your business. As I said at the beginning, we have funded 462 over our 12-year life. I'm sure that within that group, there have been a couple of stinkers, but for the most part, I think we've gotten it right. And I think you would agree with me that the ones that we've heard about today are excellent proposals and well worth the investment. So again, thank you to the presenters for sharing the results of your work with us and for doing such a fine job. Thank you to the grower cooperators that have been a part of this presentation today. And before I adjourn the meeting, Tamara, is there anything I have forgotten?
I don't believe so. However, we did receive 2 additional questions for previous presenters. If you want to take those real quick, or we can send them to them directly.
Sure.
Okay.
Let's do it.
The first one is for Dr. Dutt. I believe he is still on.
Yep, I'm here.
Okay, the question is, should we be including the Hamlin 13-32 clone in this type of analysis, as it has shown the same resistance as the Parson Brown?
We could, but our project at this time, we did not emphasize on the Hamlin clones because it was a Parson Brown project. And as I had mentioned, all the sites had very different responses to HLB from Hamlin compared to the Parson Brown. So I'm not sure if any of the sites had that clone, but definitely if we plan a field study, that's one of those clones. Plus, I'm aware of some improved clones that Jude Grosser has.
Hamlin clones, that's probably something that should be included for evaluation. Yes.
And
Thank
some
you.
of those clones I would note are going to be in the CRDF rootstock trial too. So maybe that'll help answer that question.
Right.
The final answer or the final question,
The
Tamara?
final question is for Dr. Stover. Is he still on?
I'm on the line, yes.
Any idea why HLB tolerant offspring come from sensitive parents?
It's a recombination of genes, presumably. The tolerance to HLB is likely due to dozens, if not hundreds, of individual genes interacting. And recombination of genes occurs in the process of pollen being developed and the ovule that is fertilized by the pollen, with the result that the plants that are produced have a very different combination of genes, even though they're half from each parent.
Thank you. And Mr. Dancer, that was the end of the question. I will share the screen in just a moment that will have the QR code for those who are looking for the CCA CEUs.
And the presentations will be posted on the Florida Citrus Mutual website, I assume, but they will be posted also on the CRDF website as soon as we can get all that collated. Isn't that correct, Tamara?
Florida Citrus Mutual's website is currently under construction. As soon as it's back up, they will be on there, but they will be on the CRDF website, is my understanding from Deidre.
Okay. Well, again, thank you to all of you who participated today, and thank you to the growers. Last year was tough. This next year is going to be better. Hang on if you can. Good things are on the way. So with that, Mike Sparks moves we rise. Thank you very much, everyone.